This is a step no one explains in Great Auk De-Extinction the moment the plan fails or moves. It's simple to say, brutal to do. Edit germ cells in a cousin, place them into a stand-in parent and raise a healthy Auk chick. By the end, you'll know if this step is doable now or today's stopper. This is the step no one explains, and often the moment the plan fails. Edit the germ cells in a cousin so it makes Auk-coated eggs or sperm. With that pinned, here's the claim and the real lift. The claim is seductively clean. Step 1. Build or refine a Great Auk genome from historical samples. Then identify trait clusters, streamlined body plan, dense bones for diving, wing reduction, high myoglobin muscles, cold water insulation. Step 2. Port edits into a living alcid, usually the razorbill or common murre, by editing primordial germ cells so the surrogate's gonads produce Auk-coated eggs or sperm. Step 3. Breed the chimeric surrogate pair, collect eggs, incubate, welcome a chick with Auk-like genotype and phenotype. In a pitch deck, that's 3 tidy arrows. Now the lift. Alcids aren't chickens. Interspecies germline transfer isn't routine in diving seabirds. Egg size and shell conductance differ, so do yolk composition, incubation temperature bands, and parental provisioning. You're not just toggling colour genes, you're trying to recreate a body built for deep, cold water with polygenic knobs we barely understand. The claim is a straight line, the lift is a rope ladder in a gale. Micro verdict, ambitious plan, messy reality. What's real today? We have decent Auk DNA fragments from historical specimens, enough to infer a composite genome with gaps. Candidate loci for plumage and bill keratinization exist by homology. For diving performance, we're down to proxies like myoglobin variants, mitochondrial efficiency, and limb development networks that shaped wing loading in other birds. In avian gene editing, chickens lead the pack. Stable PGC culture, CRISPR insertions, and germline transmissions are published. Quail and duck have partial playbooks, but there are no published Auk embryos, no razorbill germline chimeras, and no demonstrated trait-complete resurrection chick in any non-galliform seabird. Husbandry data for alcids exist from zoos and rehab centres, but they are thin compared to poultry. Translation: the code side is uneven but moving. The shazzy side, the actual bird we must edit, breed and rear, isn't validated. Micro verdict: pieces exist, whole pipeline doesn't. Welfare isn't a footnote here, it's the centre of the ledger. Diving seabirds are notoriously hard to keep. They need chilled, filtered saltwater with current, deep columns for submerged pursuit, and high-quality oily fish on schedule. Even then, stress behaviours spike. Feather wear, refusal to feed, and stereotypies if water temperature or prey cues are off. Now ask these same birds to act as germline surrogates. That's not egg drop-off and done. We're talking PGC harvesting or transfers, anesthesia risk, repeated handling, and a breeding regime built around a research timetable, not the bird's own. And when eggs arrive, parental provisioning is a minefield. Alcid chicks depend on precise prey size and delivery rates learned by parents in natural colonies. Hand rearing can work, but is lossy. Imprinting and dive conditioning can falter. Add transport stress between research labs, aquaria, and island pre-release pens, and the welfare tax compounds. If we need dozens of attempts to get a single viable clutch, how many surrogates are we burning through to learn? If the program can't run without chronic stress to the very birds we're using, the moral math starts redlining. Micro verdict: high welfare tax, low margin. Let's pretend we beat the lab steps. Where do young auks go? The historical range points to predator-controlled North Atlantic islands with cold, productive waters. Think Icelandic skerries, Newfoundland outliers, or rocky islets near Greenland. Modern realities bite. Invasive mammals, egging risk from humans, bycatching gillnets, oil spills, algal blooms, and shifting currents. You'll need biosecurity that doesn't blink, rodent proofing, quarantine docks, decontamination for every boat, and year-round watering. In the build-up phase, you're paying for chilled seawater aviaries, live fish logistics, veterinary specialists, telemetry, and colony site engineering to deflect storm surge. Aftercare isn't release and vibe, it's tagged cohorts, survival audits, disease surveillance, and fishery agreements that actually change net behaviour. And you must fence against a worst-case PR spiral. One storm wipes a cohort, one trawl kills 3 founders. That's public money and trust down the drain. Micro verdict? Habitat's fixable, costs are brutal. Here are the true choke points. First, alcid PGCs. We don't have a robust published protocol for isolating, expanding, editing, and recolonising germ cells in razorbill or murre. Galliform methods don't just port. Cell signalling, culture media, and migration cues differ. Second, germline transmission across species. Even if you build a chimera, will the surrogate reliably make functioning auk-coded gametes at scale? Unknown. Third, polygenic edit load. A diving phenotype isn't a single switch. Pile on dozens of edits and you caught mollicism, off-target accumulation, and developmental surprises that show up at hatch or at sea. Fourth, egg formation constraints. Yolk provisioning and shell conductance evolved for specific dive and incubation regimes. Mismatch those and embryos stall. Fifth, aquatic husbandry for founders. Raising birds that must learn colony acoustics, hydrodynamics, and foraging lanes without wild tutors is non-trivial. The bottlenecks aren't cute side quests, they are the road. Micro verdict. Bottlenecks stack, not subtract. What if the real play isn't a Great Auk, but stronger living proxies and safer oceans? For the money and ethics burn rate of resurrection attempts, you could harden burrow nesting puffins against predators, scale bycatch safety in gillnet fleets, and expand no-take zones over key upwelling. You could bankroll cold water fish recovery and island biosecurity for razorbills and murres right now. Species that actually carry auk-adjacent functions in the ecosystem. If your pitch is restore lost function, proxies already fly. If your pitch is right a moral wrong, public monuments and ironclad protections for current colonies beat lab optimism that needs a decade of animal trials. None of this slams the door on research. It just says we shouldn't mortgage living birds to chase a silhouette from a lithograph before baseline protections are in place. Micro verdict? Function now beats fantasy later for proxies. Time to call it. The blueprints exist, but biology disagrees, especially where the bird's bodies, tanks, eggs, and stress curves live. Could a lab crack alcid PGCs in 5 years? Maybe. Could a program then stack 20 plus edits, produce fertile chimeras, and rear deep diving chicks that survive fisheries and heat waves without grinding through surrogates? That's a longer shot. Meanwhile, every dollar not spent on bycatch fixes and island biosecurity is a seabird that never gets a chance to fledge. For this channel's standards, receipts first, welfare non-negotiable. The Great Auk De-Extinction gets a veto for now. Build a peer-reviewed pipeline for alcid PGCs. Demonstrate humane, low-stress surrogate cycles in a living alcid, and lock funding for habitat and fishery reforms first. Then, and only then, revisit a cautious, limited pilot. Blueprints exist, biology disagrees. If we can't resurrect the torpedo, can we supercharge its living cousins? Next episode, de-extinction passenger pigeon, rebuilding the flock cell by cell. Thank you for joining me in this episode of the series.